All right. Okay, so for our final workshop of day 2, got Xian Su from the Walter and Eliza Hall Institute. Xian did his undergraduate in bioengineering at the University of Melbourne, followed by a master's in biostatistics at the University of Melbourne, followed by a recently completed PhD at the Walter and Eliza Hall Institute. Today's workshop is on nanopore methylation sequencing and visualization.
Cool. So in the interest of time, I don't want to go too deep into all the things about methylation. Hopefully those who are in this workshop know a bit about what DNA methylation is, what it does, and why it's interesting. So what I just want to introduce is the technology that my lab and many people around the world are currently using to analyze DNA methylation, and that's the Oxford nanopore sequencing. I'm not paid by them, I swear. So the way that the technology works is it was originally produced to try and get very long DNA sequences in a high throughput, cost-effective manner. So what they do is they pass the DNA molecule through a nanopore, as you see here. And as the molecule travels through, it produces an electrical signal, which can be used in further bases. So sort of a side effect of that, because of the protocol that they were using, they didn't do any PCR. And so all of the methylation and other DNA modifications were still attached to the molecule as it passes through the nanopore. And when you do that, it actually causes a identifiable disruption in the signal. And so with this technology, you can actually get long read sequences that contain methylation information. And so with that, we'll go into my workshop. Hopefully everyone's been here for a few days and knows how to open these up. So it'll be the NanoMethViz long-read methylation data analysis workshop. And I'll just pray that this works for me. And please, nobody look at my link. Oh, no. Okay. Yes, nobody look at my link. How do I hide it quickly? What's the full screen button? Is it like F11? Who knows how I go full screen on this? Ahead of time. Thank you. Okay, so hopefully, if you've logged into this instance, you'll see that there's some data and some input folders here. So to do the analysis on this data usually takes quite a lot of time, and we don't have that kind of time in this workshop. So some of the things that we'll see in this workshop have been pre-computed. And you can run through this on your own and just delete the data folder when you have time, and you can run through everything and see that it does all work the way I claim. But otherwise, some of the data we'll see in this workshop will be pre-generated. So the first bit of code is just to download the data. So if you weren't using one of these Docker images, you can still run this workflow, but you'll just have to download this data yourself. And the data that we'll be looking at comes from an experiment where we took a cross of 2 well-known strains of lab mice. So the objective of this experiment was to try and study X inactivation and the methylation patterns in that. We won't be looking at that part of the data today. We'll mainly look at chromosome 7, where there are some well-known imprinted genes, which are genes that are only expressed from one of the parents of one of the parental alleles. So that's known to be controlled by methylation. So it's known that there are just a small handful of genes in mice which are imprinted in that they are only expressed from either the maternal allele or the paternal allele, and later on we'll see the methylation patterns of those genes. So what you basically do is, you get your samples and you just throw it into one of these long-read sequences, and if you're doing the direct DNA protocol, you'll end up being able to use the Dorado software from Nanopore to just call methylation. It'll also do base calling, but overall, I mean, I'm not a wet lab scientist, but I've been told the process is very simple. It's very straightforward, and it's much simpler than all the bisulfite sequencing procedures that preceded this. So actually, let me just, I don't think I need to see any of that or this, and I think I'll make this text bigger for everyone. Okay, maybe that's a bit too big. So the result that you'll end up getting out of the Dorado base caller, which also calls the modifications, is something called a modBAM file. And all that is it's going to be a BAM file that contains a special tag in it that also stores modification information. You don't have to worry about that. It's up to me to lose my sanity trying to parse all of that data. But what you want to construct for the NanoMethViz package is one of these modBAM result objects. And all it is it's just a collection of a path to your modBAM files, some sample annotation, and some exon annotation, which you can optionally have for annotating your plots later on. I might have to scroll up and just load all my packages in. So I'm not used to Windows controls. Yeah. So we'll just go ahead and we'll start just generating our modBAM results object. I'll just click this, I guess. So first, we just need a bunch of paths to our BAM files. And then I've put into the input folder some sample annotation. And all that is so these were neural stem cells, and I've haplotyped them. And that's the reason we have the crossing between pure strains, is that using long reads, you can actually very easily and effectively haplotype most of your reads. So using short reads actually is quite a challenge because your short reads need to overlap informative SNPs. Using long reads, it becomes much easier to do any kind of experiment where you want to haplotype. So in this case, we actually managed to haplotype, I think, over 80% of our reads into the paternal and maternal alleles. And they all come from neural stem cells. So the last thing is to grab some annotation. In this case, the package provides a whole bunch of functions. So for the model organisms I work with, and if you have your favorite organism and you want some annotations for it, please leave me a GitHub issue. But at the moment, I provide HG19, HG38, MM10, and GRCM39. So each of these, I've lost my mouse. Okay. So each of these has a corresponding function that is get exons underscore the genome you're interested in. And this just helps you grab some exonic annotation for use with the package. So most of these were taken either from Annotation Hub or UCSC, and I've just done a bit of pre-processing to get them into a nice tabular format. So you'll see that for each gene ID, there's just a number of exons that correspond to different transcripts, and they all have a gene symbol attached to them. If you wanted to use your own annotation, you just create a table that matches this format with whatever organism or annotation you want to work with. So with that, we can start making our modBAM result object, which just contains all the information that we put together. And we'll use it to plot the PEG3 gene, which is a well-known So the PEG3 stands for paternally expressed gene. So this gene is only expressed from the paternal copy and never expressed from the maternal copy. And if you scroll down, this is what it looks like. So this is the methylation pattern of the PEG3 gene. Okay, that just barely fits. So what you see here is, so there's 3 samples in each group, but I've aggregated all the samples for the groups into just one trend line. So this is the methylation trend of each experimental group that we've defined in our sample annotation. And we see that this is a gene. So if I scroll a bit further down, so this is what the exons were used for. So it provides a little track showing the gene annotation. And so the PEG3 gene is a negative strand gene, so it runs from right to left. And if we scroll a bit further back up, we see that near the transcription start site, the PEG3 gene is significantly demethylated in the paternal allele, and that's usually evidence of open expression, whereas it's highly methylated in the maternal allele, which is indicative of suppression of expression. So that corresponds to what we expect biologically of this gene. This gene's only expressed from the paternal copy and not from the maternal copy. And so in the maternal copy, it's silenced by methylation, whereas it's free to express in the paternal copy. And underneath the aggregate trends, you'll also see a heat map corresponding to the individual long reads. So this is also the sort of information you don't tend to see if you were doing short read experiments, because short reads don't tend to overlap too many CPG sites. So people just have site level data, whereas with long reads, you can actually do studies of trends along individual reads. I don't do that because I'm not smart enough, but it's something you could do, and the information is there. So if we zoom back out a bit, a lot of what my analysis does is inspired by RNA-seq analysis. And in RNA-seq analysis, we like to produce these genome-wide sample-level plots, so in particular the MDS plot. And in order to do that, I just need to perform a bit of data conversion. So rather than use the modBAM file, which is not very efficient to query at a genome-wide level, I perform a conversion into a TABIX format, which allows me to work with it a bit more efficiently. And the code is just here. So in this case, you'll see throughout this workshop, there's a bunch of these lines where if the data does not exist, then we produce it. In this case, I've pre-generated the data, so it's it should take no time to run at all. Excellent. But what that's done is it's now created a big tabular-based, compressed tabular format representation of all that methylation data, and it's the main data format that I use throughout my package. So with the modBAM results, you can do most things. So you can plot individual regions, you can plot genes, you can do a lot of plots of small amounts of the data. But if you want to use the full features of this package, you will need to convert it into the TABIX format. So once you have it in the TABIX format, you just need to make another object called the NanoMeth result object. So it's almost identical to the modBAM results, except instead of using the modBAM files, we've just replaced the methylation information with a path to the TABIX object. And just to prove that it's exactly the same thing, here we're doing the plot gene function again. So we want to plot PEG3. And now we're just using the NanoMethViz object, or the TABIX object. And it's produced the exact same plot because it was based on the exact same data. So with that, we can do some further conversion. So NanoMethViz provides a range of conversion functions that help you convert your data for use in other bioconductor packages. So for example, we can turn it into a BS-seq object, which is then compatible with DMR-seq, DSS, and BS-seq. And there's also a function that I won't show here, which allows you to convert it into something that's compatible with EDGAR for methylation analysis. So here, once again, the file already exists, so it'll just end up getting loaded in. But otherwise, if you do this at home and you delete the data object, the data directory, you'll see that it just runs the methy to BSeq on the NMR object. So we'll just run this. Okay. Do I have to address that? Ping? I hope not. Okay, so in terms of making an MDS plot, there's a bit more that we have to think about, because with gene expression, there is one value for each gene for each sample. But with methylation, there's actually two values. There's the number of measurements for methylation, and there's the number of measurements for unmethylated sites. So the strategy in order to make an NBS plot is to take the log methylation ratio, and so that combines the information of methylated sites and unmethylated sites at each loci for each sample into one value. And so that's performed by this BSeq to log methy ratio function. And all that does is it gives you a nice table where every site, well, actually in this case, if you were to do an MDS plot or a PCA plot on individual sites, it tends to be quite unstable because those values vary quite a lot. So the strategy that I'm presenting here, and you may use your own strategies, is that I've actually taken the CPG islands, and I'm going to aggregate the methylation across CPG islands. And that should ideally provide a more stable value to work with when doing these PCA/MDS plots. So that goes into this function. So I take the BSeq object, and I provide a bunch of regions. So you may choose to do this across genes or any other kind of regions that you think might better represent the differences between your samples. But in this case, I'm going to use the CPG islands, which also is produced by a utility function within my package. So I also do this, I think, for the human genome and the other versions of the mouse genome. And when you run this code, you get an MDS plot, which looks like this. So you can see that there is some differences between the paternal and maternal alleles of these samples. The difference is not actually that large, and we don't expect it to be that large because if we were to look at something like the X chromosome, we expect to see very large differences. But here, both chromosomes are active, so the differences here are mainly driven by just a small number of imprinted genes that are present. So you usually do this to get an idea of how large the differences are between the samples of interest that you're analyzing. So if you don't see any separation, if everything's just all clumped together in this MDS plot, then your hopes for good DE results are pretty slim. So now we kind of work our way down into higher and higher resolution investigations of our data. So something that we might want to do is to see if we can find some kind of signal in an aggregate of features. In this case, we are going to try and look at methylation signals aggregated over all the genes. So we take the exon annotation from our existing object, and we convert it into gene level annotation. So I can show you what that looks like. If I knew what the hotkeys were on Windows, okay. So all that's done is just collapse all of those exons into individual genes. So there's just one entry per gene, and it's just gathered up all the starts and ends of all those exons into larger regions. And what we want to do is we want to run this plot ag regions function. And for this workshop, I just took a sample of only 100 of these genes, and we want to see if we can see some kind of aggregate signal out of the genes. Take a little bit to run, but this is usually a good way to get a very broad, high-level look at your data. sometimes you might not have the coverage to investigate features individually, so you can still get something out of looking at a big aggregate of particular regions of interest or particular classes of regions. So what we see here is essentially the aggregate methylation patterns of all the genes, well, of 100 of the genes on chromosome 7. And we see that there's really not much difference between the paternal and the maternal, which is once again expected because all the genes are active in both these alleles. But what we do see is essentially the general pattern of methylation of an active gene. And let me see if I can get everything in frame. I think if I shrink this side. Okay. So what this does is it scales all the features that you put in into relative positions, and then it takes a 2 kb flank outside of those regions to help visualize an aggregate trend. And in general, what we find from this plot is that for active genes, the methylation tends to dip quite dramatically as you come into the transcription start site. But for active genes, it turns out that as you move into the gene body, the methylation quickly climbs back up. And I don't think I have enough I've already plotted more genes, but if you were to plot more genes, you'll see a smoother line. And what you would see quite clearly is that for active genes, the methylation through a gene body is actually higher than the background level. So for active genes, you expect to see a big dip in the methylation near the transcription start site, but you actually have an increase in methylation throughout the gene body. So previously, I've done analysis where I aggregate methylation across the whole gene body, and now I'm worried about all the signal that I canceled out doing that. But that's not of your concern. We'll just keep moving forward. So we can also do the same thing across CPG islands. So we just used our CPG island annotation that we collected before. And we once again do the plot ag regions function. And we'll see how long that takes to compute. Should be within a minute. I probably could have subsetted this one as well. Does anyone have any burning questions so far? There we go. So, yeah, this is just an aggregate of all the methylation signals at CPG islands in chromosome 7 for this data set. And what we see is, very concordant with what we know from literature, which is that CPG islands, for the most part, are very demethylated. And that's exactly what we see here. And we also see that it's quite a clean demethylation signal. So it's also kind of a good way to try and get an idea of how good your annotations are, because I've actually tried a few different annotations. People have tried to annotate CPG islands in different ways. And this particular one comes from the UCSC table browser. And you can see that the regions they've annotated have a very clean demethylation signal. Whereas I've tried some other annotations for CPG islands, and it's shown, a much less clear signal. So moving on from that, we want to do what I guess most people are probably interested in when they do analysis of methylation data, which is to look for differential methylation. So we want to find the regions which have different methylation signals between our experimental groups. And there are many options for this. You can use BSS, BSSeq, DSS, EDGAR. But for this particular workshop, I like to use the DMR-seq package because they actually are brave enough to provide you with a p-value for regions. It's actually quite a difficult problem to try and simultaneously discover regions and assign a p-value to them. So, for example, DSS and BSSeq don't actually assign p-values at the region level. But from what I understand, DMR-seq developed a very fancy bootstrapping technique to actually be able to provide a p-value for regions as a whole. In general, the method they go through is to call methylation or to call differential methylation at individual loci, and then they apply some sort of heuristic to aggregate all those loci together into regions. This is probably one of the most intensive parts of this workflow. So if you were to run this on real data, you should probably use quite a powerful computer. And it will take quite a long time to run, depending on the size of your sample. So once again, I'm cheating here by having all of this pre-computed. And you'll see, if it wasn't pre-computed, we just run DMR-seq here. We need to assign into our BSSeq object a condition, which is the experimental group variable, which we're just taking out of the sample annotation of our object. And what we're going to ask for here is for DMR-seq to only return regions that contain more than 20 CpG sites. It's sort of arbitrary. I've been told that this value is probably a bit too high, but it works well enough for this workshop. We'll run that. Okay. So a number of regions should now have been produced. I should have printed the regions. So the results of DMR-seq is a big table of regions. So you get a GRanges object, which contains all of the regions that DMR-seq has. These aren't necessarily the significant regions, but these are just regions that they've aggregated together. And attached to that, there's a p-value, there's a beta statistic, and then there should be a q-val. So q-val would be the adjusted p-value. So for each of these regions, they're usually marked because they contain some number of significant CpG islands or significant CpG loci. But after aggregating them and performing the statistical tests, it may turn out that as a region, they're not significant. And that's why we get these regions that have p-values that are quite high, or adjusted p-values that are quite high. So usually you get to this point, and you might send it off to a collaborator, and then they go, what I do with just a bunch of regions. So you get this data back, and what you want to do is you want to associate these regions with genes, which are much more interpretable. And the way we'll do that is using the terrific ply-ranges package. So first, we take our gene analysis. I actually learned in a workshop at this conference that I can do this a better way. Don't know if I want to try it here, though. Okay, that didn't work. Okay, so we take the gene annotation that we generated before, and we turn it into a GRanges object. And what we want to try and look for is any DMR that is within 5 kb of a transcription start site. So what we do is we take the G-ranges object, we anchor it at the 5' end, we shrink it down to a single base, and then we stretch it back out by 10,000 kbs, and the result of that should be a range that covers 5 kilobases within the transcription start site of all the genes in our annotation. Then, on top of that, we use the join overlap intersect function to overlap the annotation that we just generated for the region around the transcription start site with all of the DMR regions that were marked by DMR-seq. And the result of that should be a bunch of regions that are close to a transcription start site and also annotated with a gene symbol, which is much more meaningful. And here it is. So there are certain regions that may overlap multiple genes, which is why they will show up multiple times. But you can then look at the symbol of the gene that they overlapped over here, and now you actually have a more or less biologically meaningful list of DMRs that you can hand off to someone. I guess one last step is to then filter this table by the actual adjusted p-value. And in this case, we want to use a adjusted p-value of 0.05, the good old gold standard. And we also will drop a few columns from our table to make it easier to read. So we take the regions that we just generated, and then we select just these columns. And here we have a table of results showing genes that overlap DMR regions, and we get a actual range showing the region that's marked as DMR near that gene, and we get a barely significant p-value, but good enough. So very encouragingly, we've gone through this process and we've rediscovered the PEG3 gene as an imprinted gene. And actually, if you look through this list, most of these, there's PEG12 as well, also a paternally expressed gene. So you end up discovering a lot of well-known imprinted genes, which is very encouraging. This is also a small subset of the real data set that I used, but just enough to get significance. So with that, we want to actually visualize these regions that we discovered or rediscovered. And we can also attach the DMR regions that we found and use it to annotate our plots that we made before. So I just run this code. And so here it is again. And we have the same plot we saw before of the PEG3 gene. And now it's being annotated by a region that's been marked as DMR by DMR-seq. So I really like this because it gives me a lot of confidence when you can look into the raw data to see that I really do trust this result as DMR. Whereas often you might be able to, generate a bunch of DMRs, and when you actually visualize it, you don't really believe the result. Like it might have marked the region, but when you look at the raw data that you'll see down here, the signal is not that convincing. Likewise, you might be able to see some regions that you think, oh, that could be a DMR, but was not picked up by the statistical method. So just in the middle of this gene here. But we gotta trust the statistics. So I guess, if we just go back, we saw it before, the difference or the differentially methylated region tends to occur near the transcription start site. So that's the case for this PEG3 gene. If we look over to this other gene, let's see if we can get So the gene question is here. You can't quite read the name, but it's also a negative strand gene. And what we see is that the differentially methylated region also occurs almost exactly on the transcription start site. And the final one in our example is not quite the same. So for this particular one, you can see that the differential methylation extends almost through the whole gene body. And finishing on time. Excellent. So hopefully, this has empowered you with the confidence to try and do your own differential methylation analysis. The point of this package is to try and make it all pretty straightforward, almost as if you were doing quite routine RNA-seq analysis. It performs a lot of the data conversion. It has a wide range of visualization features. And using it, I hopefully have demonstrated that you can do a pretty robust and straightforward workflow from start to finish to discovering differentially methylated regions and visualizing them in a way that shows biological significance. And with that, are there any questions? Yes.
I don't know much about methylation data, but are there missing values? Like if we were doing whole genome bisulfite sequencing, and for many of the CPGs, we won't observe anything. And that usually creates a problem. Let's say if you're looking at a study of 300 subjects, and doing whole genome bisulfite sequencing for everyone, you'll rarely have a CPG that's profiled in all the subjects. So imputation is a real problem when you start scaling up these studies. So I don't know whether from this technology, you also get a similar phenomena.
So the way that the technology works, you don't really get missing values because as you go along a read, you will call, like the software will try and make a call on methylation at every single relevant motif. So in this case, the CG motif. So you can see here, for a particular read, it's dense along that read. It may be the case that the caller gives it a probability of 0.5, so it becomes uncertain, but the value is never missing. And you can see here essentially what the coverage tends to look like. You get dense coverage. So this is, like for this particular sample, it's only a 4x coverage in this region, but for that 4x, it's pretty well covered. I guess as a related note, I think in our data, we managed to cover something like over 98% of the CG sites in the genome that we analyzed. With the long reads, you also tend to be able to map into very difficult regions, like repetitive regions or centromeres. So there's a lot of advantages to this technology for methylation analysis.
Have you tried this with shorter fragments?
Sorry?
Have you tried this with shorter fragments, like from cell-free DNA?
Well, so you're asking if I've tried this with short-read DNA?
No. Have you tried this with shorter fragments with nanopore?
Shorter fragments, nanopore. I mean, this was a very early experiment, and the fragments weren't very big. They were around 4 kb average size. So these days, when we do just a very simple, straightforward protocol, the average length is over 10 kbs. Would you be able to describe a reason for doing shorter fragments?
If you don't have longer fragments. So I was going to say, a typical cell-free DNA fragment, 4 kb would be very long. Right? We'd assume that'd be from a neutrophil or something like that. So
I see.
I would ask my postdoc, but she's actually in the lab running some of them right now.
In principle, I don't see why this would change all that much with shorter reads. I mean, 2KB is still very long relative to everything we used to do. We used to work with 80 base pairs. So I don't imagine there's much difference. I'd love to know.
Careful what you wish for.
Hi, very cool package. I really like the aesthetics. You mentioned briefly about the repetitive regions. Do you have any experience on working with any of the ribosomal DNA sections where it's clearly not well defined whether it's methylated or unmethylated as UH?
No, personally, not. I haven't worked with ribosomal DNA. So I have looked at a lot of repeat regions, so mostly line 1 repeats. I encountered a lot of issues with that just because for this particular data set, we thought we'd been clever with the 2 strains, but it turns out that repetitive region annotation doesn't work well when it's not the reference strain, and comparison becomes very difficult. But I think we must have captured ribosomal data. So I don't know much about what makes it so special and why methylation is not well defined, but we'll have a chat about it. I might be able to look into it in my data set.
One small question. I recently had to submit a few sequencing results for Nanopore, and what I've noticed was that there's quite an abundance of non-CPG DNA methylation than I previously noted. In terms of visualization for these non-CPG, more like CH sites, in your experience, do you find it reliable using Nanopore to see these changes? Have you looked at non-CPG related DNA methylation?
I've briefly looked at some 5-HMC, so I can't comment on the reliability. Okay, so I have some experience also looking at 6MA in some bacterial data.
M6A?
And, well, it's in C. elegans data. And there's, so in terms of, so you asked about visualization and reliability of that, there's no issues with that per se. But you do need to trust Nanopore's machine learning algorithms to correctly identify the rarer types of modifications. I don't personally, I'm on the record here, I don't personally believe that they're able to distinguish the rarer types of modification accurately. And the visualization also, I mean, it tends to look quite terrible when everything is very low prevalence. You just get a flat line on the bottom of this plot. So the visualization works. I would question the actual modification calling, but it is something that technically the data that you generate can improve over time if they improve the models for calling modification on that data. So there's the silver lining there.
Thank you.
All right, thank you.
Kind of along those lines, have you played with DeepMod 2, for example, and compared it to the default Dorado output?
No, so throughout my PhD, I basically decided that I'll never use anything that's not officially Nanopore. And the reason for that is that it becomes very difficult for those softwares to sustain themselves every time Nanopore changes their chemistry. There's no real way for them to generate new training data to keep their models up to date. So yeah, that's the only reason that I don't really try any non-Nanopore based model or methylation calling.
That's a really practical suggestion, though. So, any further questions? Ember, Caitlin? No? Well, thank you, both for the practical advice and for an extraordinarily useful package.
Thanks.